from the Farm Fungi. I'm here in Denver, Colorado, and today I am going to begin my springtime experiment with cultivating mycorrhizal species, specifically ectomycorrhizal Boletus edulis. So there's a paper that I went through a couple weeks ago that describes the relationship between Boletus edulis and Cistus ladanifer. So I ended up getting some of these rock rose bushes. I got them from Dancing Oaks Nursery in Oregon, and I'm going to be inoculating these with some porcini spawn. So a few months ago I cloned a porcini mushroom and I cleaned it up on agar, and you can go through my YouTube videos and watch that whole entire process. But basically I just cloned a porcini mushroom, and now it's on grain or sawdust spawn from grain. So I'm going to be inoculating this in a mono tub, and then I also treated the plant with this Dinozyme, which clears out the root system so that I can inoculate these roots with Pseudomonas fluorescense. So I'm super excited for this experiment. It's going to incorporate plants, bacteria, and fungi all in one ecosystem, and I'm hoping that I'll be able to get some porcini fruits. So in addition to that, I'm also going to be planting some King Stropharia. So this is the garden giant mushroom, and I'm going to be using this Dr. Mike Trichivict. It's a lyophilized beneficial microbes along with the King Stropharia to see if that helps yield better fruits. So I got a sample of this stuff, and it's supposed to protect your mycelium from trichoderma, so we'll see if it works. But basically the idea is that if you spray this inoculant when you're spawning your mycelium, then the bacteria will not compete with the fungi, but they'll take up any empty spaces that trichoderma could inhabit. So before I get started with our experiment, I just wanted to let everyone know that tomorrow I'm going to be teaching an online class with Zach from Mushroom Cult. So we do online classes every so often, and it's going to be a live demonstration throughout our whole process. So check out mushroomcult.net/events, and we're going to be doing an online class tomorrow, and then we've got 2 more in-person classes for this season, and then I'm going to be start doing the farmers markets. So what's up, guys? Welcome to Fungi Friday. We are about to get started on my first run with the porcini mushrooms. So this is my first time experimenting with porcini. My plan is to do in vitro inoculation, so just within a tub, and then I'm going to scale it up to a raised bed next. So I have about 20 more pounds of spawn that's colonizing, and a bunch of more of these bushes. So I'm going to scale it up to a raised bed, and then after I do that, I'm going to scale it up to a full, 20-foot wide bed that has roses already planted. So I'm going to do some landscaping that incorporates these rock roses and porcini mushrooms. So kind of like a small, medium, and large experiment to see if there's any difference, or one of the biggest questions that is presented in all of these papers is that they're surrounded by trees and all the pine duff, and that's readily available to them, but is it going to be enough nutrients inside of a little tote? I'm not really sure, but from my experience with cultivating other saprobes, you know, I get 1 pound flushes off of 5 pound blocks all the time. So hopefully I'll be able to get some porcini mushrooms in vitro and then scale that process up. So someone asked about the Dr. Mike Trichivict, and I tried it a little bit on some more Stropharia. So I have not used this. I've heard about it, you know, from Facebook and other forums, and I feel like the idea makes sense. I just don't really know what organisms are in here, or you know, if they're even viable, but we'll find out. So, you know, I've heard really mixed things about it. Honestly, I don't know what to think. I would prefer a tea, like a plant-based tea that contains living organisms over a lyophilized product, but there still could be viable organisms in here, and that's what we're gonna find out. Alright, so the first thing to prepare this procedure is I'm just going to clean off my workstation. So this is the Dinozyme Cleanser, which I use on the roots, and this essentially will remove the biome that's already around the roots so that it will be able to accept the bacteria. So for the porcini, I'm going to be using Pseudomonas fluorescense, and this is just a ubiquitous bacteria that's in soil. It's a gram-negative with lots of flagella, so it helps to bind the roots of the plant to the mycelium. So that's really important part, and you can refer to this article here for more details about Pseudomonas fluorescens. But then we have this other product here, the Dr. Mike, which I'm going to be trying out this on the King Stropharia. Alright, so I'm just clearing off my workspace right now, and I've got some 70% isopropyl, so I'll just give this workspace a really good wipe down, and then got these Clorox bleach wipes. Alright, so normally at this point I would be spraying my bins down with bleach and letting them sit for about 15 minutes until it dries, but I already prepared these beforehand, so I'm just going to wipe them down with this Clorox wipe. And I got these bins from Max Yield Bins from Canada, so they make a really good product. I'm a pretty busy person cultivating, you know, my gourmet mushrooms, so I really like that you can order these pre-made tubs, and they already went through, you know, all the trials with the hole placements. So that's what we're going to be using today, and the most important thing for growing mushrooms in vitro is to really clean this tub out. So, you know, bleach and isopropyl alcohol, they are tried and true, and you want to make sure that you really get in the crevices, because that is where the contamination is going to hide in these crevices. So but we've got about 14 pounds of porcini spawn. So we're going to be spawning with this sawdust substrate, and then I've got a custom blend of Cottonwood and Red Oak to lay my casing layer. So oak is a really good hardwood to use for any kind of gourmet mushroom, like especially the ectomycorrhizal that are going to be really finicky. The porcini is quite adaptive, but I believe that using the best hardwoods are important in this process. So as I'm letting that alcohol dry out, I'm just going to spray off my spawn. So I got these porcini mushrooms from the West Coast, and I cloned them about 3 months ago, and I really love working with this spawn. It smells like a sweet porcini mushroom, and you can see it's got that distinct dark, it doesn't look like a typical like an oyster mushroom or anything, but it's got this nice beige tinge to it. So I'm going to be using the Dr. Mike spray on the next bin with the King Stropharia, but according to this paper, I'm just going to be using Pseudomonas fluorescens as the inoculant. So I ordered this a couple weeks ago from Ward Scientific, and then I grew it out in a liquid culture. So it's just a general broth of I think it's BPW, buffered peptone, and then I'm going to use this as the inoculant on the plants. Alright, so this bin, it looks like it's almost drying out. So I'm going to start off by breaking up the sawdust spawn. Alright, so I can really smell that sweet aroma from the porcini mushrooms. I'm just gonna grab a paper towel here and just gonna give this one final wipe down. And it's kind of critical at this stage that you don't breathe onto your tote. Alright, so now I'm just going to cut my spawn open and kind of spread it out. Alright. Alright, so I'll show you guys a trick that I use sometimes for inoculation, and you can take this bag and kind of flip it out so the inside, the sterile part, you can use to kind of spread out your spawn. And you can see some really nice rhizomorphic pieces of mycelium, and it smells like fresh porcini. Alright, so that was about 7 pounds of spawn, and I'm gonna add the second bag now. And I'm going to do the same thing with the bag. So I'm preventing touching the substrate with my gloves or any of my skin, and I'm only using the inside of that bag, which was sterile. So I used an oak sawdust blend for the porcini, and it really makes like a nice, almost like a soil for these plants. So I'm gonna just kind of dig out some spaces to plant these rock roses. So this is a rock rose. It's a Cistus ladanifer, and I got it from Dancing Oaks from Oregon. So about 5 days ago, I received it in the mail, and I sprayed it off with some neem oil to mitigate any kind of bugs that might be associated with this. And then I used this Dinozyme to flush the roots. So this should essentially be sterile or very minimal biology on the roots, and it came with this straw so that it held the moisture in. But you can see it's got a nice healthy root stock, so I'm just going to be really careful here. And I mostly chose to do 2 plants as an insurance policy in case one of them didn't make it. But honestly, I don't know if it needs or if it benefits from 2 plants over 1 plant, but I kind of just wanted to do 2 of them to start with. Alright, so that will probably be the only time I ever bring a plant into my lab, but I feel like they were pretty clean. I used neem oil and this enzyme to kill off any microbes. Alright, so now I'm going to switch my gloves. And we are going to inoculate this bacteria. Alright, so I grew out the Pseudomonas fluorescens in this liquid broth, and it doesn't require much concentration of organism, but I'm probably going to use about 20 cc's per plant, So that way the bacteria will be able to fuse the mycelium from the porcini to the plant and create that ectomycorrhizal relationship. So I just have to grab a syringe real quick. So I have this glass syringe that is 20 cc's. So I'm just going to be using a 16 gauge needle. And then I'm just going to draw up the 20 cc's. Alright, so there should be Pseudomonas fluorescens in here, and I'm just going to inoculate the roots of these Cistus ladanifer plants. Alright guys, so that should do it for the porcini. Inoculation, now I'm just going to case it with some wood chips and a little bit of sawdust. So it's really important to have a blend of different sizes of wood chips. It's going to create variations in the mycelium, which would help trigger pinning. So I just want to point out that, I want to give a lot of credit to the people who wrote these papers, because all I had to do is do my research and read them, and they did a lot of the hard work up front. But there's a very small chance that this is going to pay off. But I feel like if I can do it in my lab, or encourage someone else to pull it off, then that's just going to lead our industry even farther. So that being said, I will case this mycelium and then move on to the King Stropharia. So for those of you who just joined in, I think I'm going to scale this up next to a raised bed, and then I'm going to do it in my backyard along a back fence that gets a lot of shade. So this is just the in vitro portion, which just means it's taking place inside of a controlled environment. So I feel like there's a lot to be gained, and it's always been a dream of mine to be able to cultivate porcini mushrooms. They're definitely my favorite mushroom. They have like a really sweet flavor if you've never had a porcini. And one of the problems is that when you find them in the wild, there's always, bugs or worms in them. So, the benefit of cultivating versus finding them in the wild is that there's going to be less bugs. And hopefully you can get them whenever you want during the time of the year. Alright, so I just I sterilized this for about 6 hours, and then I let it cool down to room temperature. So that's another important factor, is you want to make sure that everything's cooled before you do your inoculations. So this is a blend of Cottonwood and Red Oak. The Cottonwood is a softer hardwood, so I believe that it will colonize a little faster. And then the Red Oak is just, you know, a really common hardwood that a lot of species adapt well to. So you want about an inch of casing just to maintain that moisture content. And this one does not, this mixture does not have any nutrients, so it's not as important to be sterile at this point. But I'm just making sure that the substrate is below these air holes. And then I've got some painter's tape that I'm going to cover up the holes until the substrate fully colonizes, and then I'll switch this out for some micropore tape. So I like using the painter's tape because it still has a little breathability and it doesn't leave like a sticky residue. All right, so then the final step is just placing your lid. And once again, I'm just going to give this a quick spray down. I cleaned it out really thoroughly right before I started the video, but it doesn't hurt to probably the most important part is just cleaning. Alright, and then these Maxio bins comes with little handles that locks the lid into place. Yep. Except for that one. And then I'll just go ahead and tape up these seams. For you, those of you guys just joining in, I'm going to be doing a class tomorrow with Zach from Mushroom Cult. It's an online Zoom, and we're going to go through all of our procedures for cultivating mushrooms and breeding mushrooms. So if you're interested in that, it's going to be tomorrow from 9 until noon, and you can get tickets at mushroomcult.net/events. And we're going to do probably one more online class, and we have 2 more in-person classes until the farmers markets start. All right, almost done with the porcini. So after this is all done, I'm going to just place it in ambient light upstairs, just room temperature, and a southern facing window, but indirect light, just so it gets enough sunlight to keep the plants healthy. But Alright, so that one's all labeled up, and I'm a little bummed out about this broken handle, but it'll work. So next, we are going to inoculate the King Stropharia, and I wanted to test out the Dr. Mike trichoderma prevention. So alright, I'm just going to clean this up a little bit. Alright, sorry about that. Just getting some things in order. For the next tote. So I've got, once again, it's one of the max yield bins. I may have misplaced that and find the, there it is. Alright, so I've got the Dr. Mike Trichoderma Fix, or Trich Evict, and I'm going to be utilizing this along with the King Stropharia. So one of the problems with trying to grow Stropharia rugosoannulata in vitro is that it requires companion bacteria, or it's very susceptible to trichoderma. So this is supposed to help fight trichoderma by providing those beneficial microbes. So it looks like you just add 2 grams to a liter. So this is just water from my, it's just filtered water. So someone asked about the root cleaner. So all I did was use this Dinozyme, which is an enzyme cleaner, and I flushed the roots about 5 days ago. So the idea is that this would clear out any biome, and then it's just an enzyme, so it will break down eventually. And you know, I just inoculated with Pseudomonas fluorescense, which is a really rigorous bacteria, and my hopes is that that will just overcome these enzymes. They're about 5 days old by now, and I kind of flushed it out just with water, so it's very dilute, and the idea is that it killed off any microbes, which would allow for the space for the Pseudomonas. So it doesn't really say what ingredients are in here, but we can open it up and take a look. So it's just a gray powder, and I'm gonna take about a half a gram and just put it in here. There's about 500 milliliters of water, so I'm just gonna guesstimate. Alright, just gonna shake it up real good and let this hydrate while I finish cleaning out my tub. Once again, you just want to really get in all the crevices. So that is nice and clean, and it's ready for the spawn. So I just used oats as my spawn, and then this is just hardwood pellets. So you can see it's a lot thicker than the porcini. But you can start to see some of the grains coming out. And I've tried to fruit Stropharia inside of a bag, but it never pinned, and I believe that it's because there wasn't the proper organisms that induce pinning in like an outdoor bed. So there's about 5 pounds of spawn in that. The Stropharia spawn is a lot more rigorous. The spray is probably hydrated now, so I'm just going to give it a couple doses of the Dr. Mike. It's pretty odorless, and it's almost like a creamy color. Go ahead and mash in this spawn. Yeah, so someone just asked about the lobster mushrooms. So I haven't thought about that yet, but it might be a really cool project to try to parasitize like an oyster mushroom with a lobster mushroom. But right now I'm just focusing on these beds so that I can scale this up outside. Alright, so once again, I've just got some sawdust. Spread that as a casing layer, and then I'm going to do one more final mist. So I definitely didn't need 400 microliters, probably, 20, but it seems like it plugged up my sprayer. So one thing to note on this Dr. Mike is you might need like one of these more industrial sprayers. I like this one though because it's like a fine mister. But yeah, so someone asked how much the max yield bins were. I don't really remember. I got the 4 for like 150 something because it's free shipping, but they're really well made except for that one that just cracked. But I like the ease and the volume of it. I feel like this one is going to work perfectly for King Stropharia. You know, it's got the head space required. Yeah, I'm just pretty busy these days, so you can go out and, you know, just drill some holes and make your own, but I think it's cool to support a really cool company. Alright, so then guys, I'm very excited for this project. I think there's a few more liquocultures of porcini left on Etsy, and then as soon as I get some fruits out of this culture, I'll probably post some liquocultures of the King Stropharia. I'm just going to seal up these holes until it colonizes, and then once I start to see some mycelium poking up, then I'm going to switch to micropore tape. So I'll keep you guys updated, and then as the weather heats up outside, I'm going to move these projects to raised beds, and then I'm also going to do some ground inoculation. So I like to do a pretty large vegetable garden, and I'm pretty excited to add some more fungi to help with the plants. Still a little bit too wet. There are some companies that make these like little magnetic discs, and those would be really helpful right now, but I like this tape because it doesn't leave any residue. So hello from Germany. So for all of you guys just joining in now, we did some porcini inoculation with some Cistus ladanifer plants, and we're just finishing up here with a King Stropharia. So I'll keep you guys posted on these as they progress. And then as it gets warmer outside, I'm going to be moving these into some raised beds. I got these really cool fabric raised beds that, you know, I believe that they're going to be really nice for growing mushrooms because they have this waterproof layer in the middle, and typically like the fabric beds will dry out really quickly, but it's nice because when you water it, there's still enough aeration for the mycelium. Alright, so there you have it. We've got our porcini mushrooms with the plants up top, and then we've got the King Stropharia with the Dr. Mike Trichivict. So hope you guys enjoyed that video. Give us a thumbs up. Subscribe if you haven't subscribed yet. We're going to be doing a lot of these experiments in the future, and you don't want to miss out. I'll keep you guys posted on these, and I hope you guys have a good weekend. It's supposed to get a lot of snow here in Denver, so I'll have these to look after over the weekend. Alright guys, thanks for joining in. Happy Fungi Friday and much love.